At this point, you know how natural selection works, and you should be able to predict how it would cause the population to change if selection were favoring one genotype over the other. We'll be reinforcing this with our lab activity this week. Right now, it's time for us to think about randomness in nature. Here, the specific kind of statistical randomness resulting from sampling in finite populations. There are no biological populations that are infinitely large. The closest thing to an animal population of size infinity is krill, little shrimp-like things that whales eat. But even in the krill species, Euphausia superba, which totally smokes the human population of n equals 7 billion, that's 7 times 10 to the ninth. The population size for the krill is something like 10 to the 14th power. A big, big number, but not infinity. Now, krill are like humans and the majority of all sexual organisms in the way that each krill has one mommy and one daddy. I wasn't able to Google much on krill reproductive biology, but if we were to presume the same kind of XY sex determination we're familiar with, you would expect that exactly half of the 10 to the 14th krill to be born female, right? That would be exactly 5 times 10 to the 13th female krill to 5 times 10 to the 13th male krill, exactly. Now, if you were to stay up late one night and count all the boy krill and the girl krill in the world's oceans, and you were to find 5 times 10 to the 13th exactly male krill, but 5 times 10 to the 13th plus 87 females, that 87 would be a departure from your expectation of exactly half male and half female, right? Now think about this. If you were to measure the allele frequency for a particular gene, little a, in the krill this year, and you find it to be exactly 0.25, that's Q equal to 0.25. And then next year you come back and measure it again, and you find it to be 0.2499999. In other words, just a skosh off of exactly 0.25. This would be the same kind of thing, by which I mean a departure from the expectation of Q equal to 0.25, but likely caused by the same kind of randomness that caused the departure of 87 extra females from our expectation of exactly half male and half female. Now, it would be kind of silly to say that either the excess of female krill babies or the minuscule amount of allele frequency change required some kind of special cause favoring females in the first instance and the big A allele in the second. And you might even offer a way of testing this. If it's randomness, then a slight departure favoring females isn't something you would expect to happen consistently. And so if you were to count up males and females every year for the next 10 generations, you would expect an excess of females half the time and an excess of males half the time. The likelihood that you would end up at exactly 50% is really low, right? So you expect there to be some deviation from 50%, but you can't predict the direction. You can't predict if it's going to be an excess of males or an excess of females. Now, smaller deviations are going to be more likely than larger deviations. Exactly 50% of each sex would actually be the most likely outcome, somewhat more likely than either 51% female or 51% male. 53% female would be even less likely. If you're actually counting up 100 trillion krill as your sample size, the likelihood of being as far off as 55% rather than the expected 50% just by chance, this would be astronomically unlikely. But if your sample size were only 20 instead of 100 trillion, think about it. 11 females and 9 males would not be surprising at all. Let's say that again. Getting a result of 55% rather than the expected 50% would be extremely unlikely to occur by chance in a sample of 100 trillion. But it would not even cause you to bat an eyelash if the sample size were 20. This is getting at another important dynamic of random genetic drift. The smaller the sample size, the more likely it is that random chance will result in a larger deviation from the statistical expectation. Random genetic drift has a larger effect on smaller populations. In a population of size 100 trillion, drift will have little effect. But in a population of size n equal to 20, randomness will cause significant evolutionary change due entirely to this chance sampling effect. Applying this same logic to changes in allele frequency now, if the population starts out at exactly q equal to 0.25, the expectation in the absence of drift or any other microevolutionary force is that it will still be exactly at q equal to 0.25 in the next generation, right? And yet one generation later, we find that it's 0.20, a shift of delta q equal to negative 0.05. In a population of size n equal 20, this could totally happen as a result of random chance. You wouldn't be surprised by this at all. It's not something you would want to write up and submit for publication. But if the same amount of gene frequency change, delta Q equal to negative 0.05, were to happen in a population like krill, where n is equal to 100 trillion, the chances of this happening by random chance would be so remotely unlikely, as in winning the lottery 3 weeks in a row unlikely. It would have to have been caused by something other than chance, and this is a result that would probably need to be reported. If you're following me on these examples, you have the basic idea of random genetic drift. One key aspect of random genetic drift is that it's, well, random. Increases and decreases of allele frequency occur with equal likelihood. Smaller deviations from the expectation are more likely than larger ones. The second key dynamic is also super important. The effect of random genetic drift becomes greater as n becomes smaller. In populations that are small enough, genetic drift impacts the gene frequency and the change can be significant, even swamping out the effect of weak selection. Conversely, in populations that are sufficiently large, genetic drift can be more confidently ruled out as a cause of any substantial evolution. This is the user interface of the PopG simulation we'll be using in the lab. The PopG settings allow you to set the values of W, that's fitness, remember, for the 3 genotypes, big A, little a, and little a. And in the default setting, they're all the same, 1.0. Think of these as our snapdragons, red, pink, and white. No flower color has an expected rate of success that's higher than anyone else. No natural selection. Now, in a large population, set n equal to 2000, which is about as high as is allowed in the simulation. The amount of change you see over 100 generations is relatively little. But in a smaller population, here n equal to 20, the fluctuation from one generation to the next is much greater, such that at this point right here, around generation 60, the line hits the top and stays there. At the top of the graph, P, the allele frequency of big A, is equal to 1. And that means all of the genes in the population are big A, and there are no little a genes, and so everyone in the population is big A, and all of their offspring will be big A, and the population will have to remain at P equal 1 forever, or until a different allele is introduced, and this could happen either through mutation or gene flow, which we'll discuss shortly. Now, if all the genes in the population are big A, how much genetic variation is there in the population? Well, as far as the A locus is concerned, everyone has the same genotype, big A homozygous. And so they're basically genetically identical clones. Variation is non-existent. When we started out, P was equal to 0.5. And so there was half big A alleles and half little a alleles. Genetic variation existed. Genetic drift started with genetic variation and ended up at P equal to 1.0, and the allelic variation was no more. And here we come to the third important dynamic of drift. Random genetic drift causes the loss of heritable phenotypic variation. The ultimate outcome of drift is the fixation of the population, either at P equal to 1.0 or P equal to 0.0. Just as easily, our population could have drifted down to the bottom, in which case it would be stuck with all little a homozygotes. Either way, stuck at the top or stuck at the bottom, this loss of genetic variation is irreversible. Even in our bigger population of n equal to 2,000, or for that matter, n equal to 100 trillion, genetic drift causes the gene frequency to nudge up and down ever so slightly, but given enough time, it will end up either on the top or the bottom. Now, let's think about the ramifications of this for real-world populations. Which of these two species is likely to possess the greater amount of heritable phenotypic variation? Species A, whose great numbers make it a species of least concern when it comes to the different levels of conservation. Or species B, that is critically endangered because there are so few of them. So yeah, easy, right? More variability in the species of least concern because in the critically endangered one, random genetic drift will have caused much greater loss of allelic variation because of its smaller population size. Now, which of these same two species will have a greater ability to evolve and adapt in response to future changes to their habitat? Now, there are a couple of more evolutionary forces we should get to before our lab exercise: mutation and migration, aka gene flow. Mutation is what it sounds like, the physical changing of one allele into a different allele, presumably due to some error in DNA replication, something occurring at a molecular level and totally at random. DNA replication is a purely mechanical process. It isn't concerned with the information that's being copied. It just goes letter by letter, matching up T's with A's, C's with G's, for all of the 6 billion bases in your diploid human genome, every single cell generation. Doing this billions of times as we do, there are going to be some mistakes, and sometimes those mistakes will occur in the cell lineages that are transmitted from one generation to the next. If one such error caused a big A allele to be converted into a little a allele, it could result in a big A little a heterozygous baby being born to two big A homozygous parents. And this would represent a very tiny, but still a non-zero change in the population allele frequencies, caused not by selection, not by drift, but by mutation. And it is in this sense that mutation is a microevolutionary force. But realize that DNA replication is almost an error-free, foolproof process, and an actual mutation is so unlikely and rare, between 1 in a billion and 1 in 10 billion. This couldn't possibly be of much importance in microevolutionary change, right? Yeah, good catch. Any amount of change in allele frequency from generation T to generation T plus 1 resulting from any of the other microevolutionary forces will be far, far greater in magnitude relative to mutation. But you have to think about mutation in different terms and on a different scale of time. Mutation is actually an extremely important microevolutionary force. How can this be? Well, for one, in the total absence of mutation, the heritable phenotypic variation in a species would eventually erode to nothing. Given the finite population size of a species and random genetic drift alone, we'd all eventually become fixed for the same alleles across the entire genome. And at that point, we'd all be genetically identical. Now, if you add to that some natural selection favoring one allele or the other, this loss of allelic variation only happens that much more rapidly. And once we lose all allelic variation and we're genetically identical, there's no heritable phenotypic variation and evolution grinds to a halt. Why doesn't this happen? Mutation. Here's another way to get this perspective. If you have allelic variation for some phenotypic trait in your species today, that means that at some point in the past, a mutational event caused the conversion of one allele into the other. By at some point in the past, we could be talking about last year, but Probably not. We're really talking about any time in the past, and this extends to well before the existence of the species that we have today. Take the example of allelic variation for ABO blood types in humans. You might know about the three alleles. Two are codominant and one recessive. And the result of this variation is different antigens on the surface of red blood cells, and you have to know this in order to do things like blood transfusion. You might be type A, type B, type O, or type AB. And if you don't know your blood type, you should find out. You know who else has these same ABO blood groups? Chimpanzees, gorillas, orangutans, gibbons, even some old world monkeys. Now it's evident from this that the mutations causing this allelic polymorphism predate the split between monkeys and apes. And this means sometime over 20 million years ago. So if our species has X amount of heritable allelic variation presently, it's because of this rare introduction of new mutant alleles adding incrementally to genetic diversity over tens of millions or hundreds of millions of years. Earlier, we were talking about species endangerment and the effects on allelic variation and the ability to evolve in the future. If a once numerous species becomes critically endangered over a period of many generations, and then it recovers, once again to populate nature with great numbers, will they automatically be regaining the same level of genetic diversity that they possessed before their numbers were cut to a small number during that period of endangerment? Northern elephant seals. So if you get in your car and drive north through Santa Barbara and on up PCH through San Luis Obispo County, you're sure to come across some beaches that are filthy with seals, seals that look like gigantic garden slugs. The biggest of them are the territorial bulls that are up to 10 times more massive than the cow seals, which are probably most of what you see. Ah yes, the northern elephant seal. In the 1800s, these pinnipeds were hunted to near extinction for their oil. Basically, you kill a seal and then you melt the fat off the body so you can have fuel for your lamps and lubrication for your sewing machines. These seals were hunted and hunted until they were totally extinct in the United States and Canada, with only a few managing to survive in Mexico, probably fewer than 100 individuals. But people still continued to hunt them. Got to get some skins to put in museums before they're all gone, right? And yet somehow they persisted. And it wasn't until after 1920 that the seals were protected by law and their numbers began to recover to their current of least concern status with over 150,000 individuals. Not all of them are on the beach at San Simeon. The range extends from Mexico to Alaska. Yay, seals. Now, despite the fact that the northern elephant seal population is strong today, its genetic diversity is very low as compared with, say, the California sea lion, which has about the same range and current population size as the elephant seal, but it never underwent the population bottleneck. Maybe the fat is not so good for lamp oil, and that's why they weren't hunted. Okay, so it's been about 50 years since the elephant seal population has been large again, but the allelic diversity that it once possessed had been the result of mutations accumulating as very, very rare events over 50 million years, and who knows how many different species of mammalian ancestors that eventually gave rise to the pre-bottleneck population of the northern elephant seals. Most of that variation was lost during its period of very low numbers, the population bottleneck, and the rampant genetic drift that goes along with very small population size. Now, if these seals were to continue to exist for long enough, mutation could theoretically restore allelic variability, but this process would likely take tens of millions of years, far longer than the 1 million years that most species have as their time on Earth. Mutation is, ultimately, the only source of new allelic variation for a species. When a species gives rise to a descendant species, it passes along much of the mutational variation that occurred and accumulated during its existence, as well as the allelic variation that it received from its predecessors. Without mutation, heritable phenotypic variation would dry up entirely, and there would be no further evolution. For now, this is how I want you to think about mutation, okay? Let's talk a bit about how many people, biologists included, mischaracterize the way that mutation contributes to evolution. The popular talking point is that when a mutation happens to be favorable, it leads to natural selection, causing that variant to become the standard for the species. You've probably heard this before, and there's actually some truth to it, but it isn't adequate. For example, this emphasis on favorable mutation completely fails to account for mutation's role in maintaining heritable phenotypic variation over the long run, as required in order for populations to continue to evolve and adapt. When one allele mutated into another in the distant past, was this necessarily a favorable mutation? If it was, then why didn't it completely replace the original allele? For a far richer and more accurate picture of mutation as an evolutionary force, you want to remember that it's the cause for the full array of allelic variation in any species genome. All the genetic diversity in humans, for example, is the legacy of mutational changes over the multi-species history of our ancestral lineage. The last evolutionary force we'll talk about in this lecture is gene flow, migration, same thing. What happens when one population exchanges migrants with another population? Well, unless the migrants being exchanged were identical, each of the populations becomes a little different than it was before the exchange. If my population of snapdragons with p equal to 0.75 were to receive either migrant seeds or migrant pollen that bring in genes from another snapdragon population, then unless the incoming genes exactly match those in my population, this will cause a change in my population's allele frequencies. If I send migrant seeds and or pollen to the other population, then its frequency will change. In a very limited way, gene flow is similar to mutation in the sense that it provides a way by which a population that's stuck in a completely homozygous state can be rescued back into a state of genetic variation with the introduction of the other allele. For example, if my population had become fixed for red flowers, then gene flow could restore genetic variability if it were to reintroduce some white flowered genes. But migration is also very different from mutation. For example, unlike mutation, gene flow actually has the potential to cause a great amount of short-term change. The influx of new genes from other populations could have a major impact on gene frequency. Also, unlike mutation, the genes being introduced by migration represent nothing new as far as the species is concerned. Mutation, on the other hand, brings in brand new, never seen before allelic variants into a species, and migration does nothing of the sort. We'll come back to migration when we get to the topic of speciation. For now, I'm happy if you can explain back to me how it qualifies as a cause of microevolutionary change. To recap this lecture, we discussed 4 microevolutionary forces: natural selection, random genetic drift, mutation, and gene flow. Each one earns its claim as a microevolutionary force because it can be the direct cause of a population changing from one generation to the next. This change can be characterized in terms of the phenotypic distribution, as we did in describing and contrasting the directional, stabilizing, and disruptive forms of natural selection. Alternatively, we could characterize the population in terms of allele frequencies and genotype frequencies. This is the approach of population genetics, in which case the evolution across a single generation may be cast simply as delta p, the allele frequency in generation t plus 1 minus the allele frequency in generation t. With random genetic drift, the evolutionary change is an unpredictable deviation from an expectation of no change, and it's due to the sampling effect of a finite number of individuals. Genetic drift is more pronounced in smaller populations than larger populations because smaller populations are subject to greater relative amounts of these chance sampling effects. Genetic drift also causes the loss of allelic variation, which generally has a negative effect on a population's ability to evolve into the future. And this is a major cause of concern to conservation biology. Mutation has effectively no measurable impact on populations from generation to generation, and yet it is ultimately a very, very important evolutionary force because it's the only source of novel allelic variation for the species. Gene flow specifically concerns situations where there are different populations of the same species that exchange genes via migration. In the next lecture, we're going to walk through a hypothetical population in which none of these four forces are occurring. No natural selection, no random genetic drift, no mutation, no migration. Basically, if you eliminate all possible causes of microevolutionary change, you'll have a non-evolving population. And if this non-evolving population is also undergoing random mating, then there's a very special mathematical relationship between allele frequencies and genotype frequencies. And I am referring to the Hardy-Weinberg law. We will also be spending a bit of time in the next lecture on some philosophical type concerns, specifically the boundary between science and non-science. We talked about this in a previous lecture. Remember the science of uniformitarian explanations for geologic features versus non-science, Cuvier's supernatural explanation of catastrophism, and the non-scientific natural theology of William Paley to account for life's apparent design versus the naturalistic, that is, scientific explanation offered by Charles Darwin, which is evolution by natural selection. Well, these conflicts really haven't gone away. I've posted a documentary that I'd like you to watch in advance of class time, before viewing the next lecture video. It's about an attempt by Christian fundamentalists to promote a form of non-science called intelligent design, and to do so in the classrooms of public high schools in a school district in Pennsylvania. This attempt was shot down by the courts, and this is the happy tale told in this video. But sadly, this same hijacking of public science education has occurred again, and it has been more successful in subsequent attempts in other parts of the country. There's a lot of bad science out there. Heck, for all I know, you might be an intelligent design creationist yourself. I just want to spend some time on the front end of the next lecture, making it clear where I see the boundaries of science education. Hint, it does not include anything supernatural. But anyways, the link for the Vimeo hosted video is in the class schedule. It's pretty easy viewing, much more so than my videos, sorry, and it has a runtime of just under 1 hour.